Hi, welcome to this lecture on mathematical methods for biologists. Today's topic is diffusion equation. We will discuss about diffusion equation and we will answer the following question. We will answer the question, how does concentration change with time at different locations due to diffusion? So, this is a question that we will answer. How does concentration change with time at different locations due to diffusion, and what do we mean by this question? Let us understand this a little bit, and that will give us some idea about what are we talking about. So, if we look at a cell, the concentration, if you look at a cell, the concentration at different places could be different. For example, the proteins would be very high concentration at one end and then very low concentration in the middle, and there could be very high concentration here. So, the concentration will first, if I plot concentration versus the x distance, it could decrease and then somehow increase. So, the concentration could change in space according to this fashion. So, at different locations, so by location I mean space. Location means space. So, that is at some point, at some region. So, given x, y or z in 3D, in 2D, given an x, y position, in 2D like x, y, z position, we would get different positions and different concentrations. So, the concentration C is a function of x, y and z. It is also a function of time, because things should diffuse from here to here. There could be a diffusion from a higher concentration to lower concentration. There will be a diffusion from here to here. So, the concentration here would increase after some time. So, the concentration increase as a function of time also something that we need to think about, and we can get C of x, y, z comma t. So, the real concentration C would be a function of x, y, z and t. In 3D space, x, y, z represent the 3D space and t represent the time. So, as a function of the time, the concentration will change. For example, if this is the concentration at t equals 0, after some time the concentration will come down here and the concentration would increase. Something like this could happen, for example. So, the concentration profile will change as the protein molecules or the molecules of our interest diffuse from one place to the other. So, given that there is diffusion, how does this concentration C change with x, y, z and t? That is the question that we would ask. So, what are we going to ask? We are going to ask the following question. Given that there is diffusion, given that there is diffusion, how concentration changes with x, y, z and t. How does this change as a function of x, y, z and t? In other words, the question is, what is this function? Can we calculate this function C x, y, z comma t given the diffusion? How do we calculate it? Can we get this function if we know some initial boundary conditions? Can we get this function diffusion? What is the equation that will be obeyed by this C is the question that we would want to address. Now, what do we know? We know that the flow, the current is something that we have already computed. So, what we know is that if there is a concentration gradient, if there is high concentration at, so if there is high concentration at one place, so there is a very high concentration here. so there is high concentration and very low concentration at these locations, we know that there will be a current J, and this current is given by minus d in one dimension dc by dx x cap. So this is the thing that we know. Since c is a function of x, y, z, this is J is actually, the diffusion current is actually minus d del c by del x x cap. So this is something that we already know. Now, given this J knowledge that we have, what do we know about the change in concentration? So let us think about this. Just to remember that J is a function of x. So if you have different concentration, the J would be a function of x. For example, if you have a tube like this, and if you have a lot of molecule here, and very few molecule here, and a lot of molecule here, and very few molecule here, and a lot of molecule here, the current here would be very large because there is a lot of molecules, so they will flow, while current here would be very small, current here would be large, current here could be small, and current here could be large. So J at this x point would be different from J at this x point, would be different from J at here, J at here. So J at different positions of x would be different. So J is a function of x. So this is something that we know. Now, let us think of J. What is J? J is the flow. So if the flow at this point and this point, let us consider these 2 points. So let us consider a small, let's consider a tube like this, and we are marking 2 points, x and x plus delta x. These are the 2 points that we are considering, and there are more protein molecules everywhere which we are not drawing. That is, there is a lot of protein molecules. Okay, let me mark protein molecules everywhere with some concentration. I don't know what is the concentration. There is some concentration and there is some protein gradient. I am not saying anything about concentration. I am only saying that if the flow here, let us assume the flow at x, if the flow at x is along this direction, so let us draw the flow at x. Let us say there are 300, I drew 3 arrows, 300 protein molecules cross every second this line, and here also 300 molecules cross every second from here to here. So these 2 lines, 300 molecules, let us say, are crossing this line in every second. That is the flow here. The flow here is also 300 molecules crossing every second. That means the flow here and the flow here are the same. So the J at x is same as J at x plus delta x. So if we have J at x is same as J at x plus delta x, so the 300 molecules are crossing this line every second. So that is the flow here. 300 molecules are crossing this line every second. These are the flow here. If they are equal, if the flow here is equal to the flow here, in this in-between region, 300 molecules are coming in, 300 molecules are going out. Therefore, the net number of proteins, net concentration of the protein will remain unchanged because whatever came in went out. So the flow at 2 nearby locations are equal, the concentration in between will not change. This fact, if we write mathematically, that becomes the first part, that becomes an interesting equation which we will see how to write this. So the physical fact we are writing is, let us repeat, we have a cell like pipe or a tube in which there is a concentration gradient of this red particle proteins, protein particles, and I am taking 2 points x and x plus delta x, and I am saying that at x, if I look at it, I am finding that the flow is along the plus x direction, and the flow is some number which is let us 3 arrows, which would let us say this means 300 protein particles are crossing this line every second. That is the flow here, that is j of x. J at x plus delta x, let us assume it is exactly the same as this in the same direction. That is, here also 300 particles are crossing in the plus x direction. If this current is equal to this current in both direction and magnitude, the concentration of the red proteins in between will remain unchanged. So the concentration here will be 0. So that means will not change with time. That means 300 particles came in, 300 particle went out. So if this is equal to same, this implies that the dj by dx is 0. So this would imply that del j by del x, the change in flow j of x plus delta x is equal to j of x, therefore the derivative will be 0. If these two are equal, there is no change in flow as we go along the space, therefore del j by del x is 0. The assumption here is, of course, is that these two are very close to each other, delta x is very small. Then I can write del j by del x is 0. So if del j by del x is 0, that means the flow here is same as the flow here, then the claim is that the concentration will not change with time. The concentration here will remain constant over time. So let us write that. So let me write this once more. Let me draw again. We have this. We have these 2 points, x and x plus delta x, and we are worried about the flow here, j at x and flow at x plus delta x, and we are saying that the flow here is exactly equal to the flow here, equal in magnitude, equal in direction. Therefore, j at x is equal to j at x plus delta x. This means the derivative is 0. This means del j by del x is 0. And because of that, if same number of particles come in and same number of particles go out, here the concentration change with time. How does the concentration change with time? This will be 0, because this will not change either. So del c by del t will be 0, del j by del x will be 0. So this is the first point that we want to make. The second point is, let us imagine a case where this is not equal to this. So that is the second point that we will consider. So let us consider again a tube like cell. Let us consider 2 point x and x plus delta x, and the flow here x, and this is the flow at x plus delta x. Let us say the flow at x is 300 particles in every second and only 100 particles go out. So there is a flow out is only one line. That means this is less compared to this. So del j by del x is not 0. This, whatever comes in, lot of particles are coming in, 300 particles are coming in, but only 100 particles are going out. What does that mean? That means here the proteins particles will accumulate. This means that protein particles or protein molecules will accumulate at this point. Accumulate. There will be an accumulation of protein particles because there is a lot of protein particles are flowing in and only few are flowing out. So the concentration change here, del C by del T, will not be 0, it will be a non-zero, will not be equal to 0, it will be a quantity, and this will not be 0. This is we know because the concentration will change with time. As we wait, more and more particles will diffuse in and they will start accumulating here and the concentration will change with time. Del C by del T will not be 0. what will del C by del T be? And the claim is that the del C by del T will be equal to del J by del X. So let us look at del J by del X. So there is a del J by del X is also not 0. So if I look at del J by del X, this is also not 0 because there is a higher here and lower here. So the J changes with X. J is decreasing with X. So del J by del X is negative. And the claim is that del C by del T magnitude is minus del J by del X. This is the claim. This equation is called the diffusion equation. This equation is also called a continuity equation because the only thing we said here is whatever comes in goes out or not. That's the only thing we checked. We checked essentially the continuity, and this continuity will give us this phenomenon of diffusion, will describe the phenomenon of diffusion, and this will give us diffusion equation. So this is the magnitude. We will worry about the direction later. This is a scalar. This is a so if I put J is the magnitude of the vector, and I only want to put the magnitude here, we will worry about the direction later. So this is the magnitude, and now let us think about this a little bit more carefully. What does this mean? What is this del J by del X? So here we can see that J at x plus dx minus Jx. So the flow here is less compared to the flow here. So this minus this will be a negative number. So this will be a negative thing, and this would mean that the del C by del T is a positive thing, because negative, and del J by del X is negative. So this will give us a positive number. So let us look at it a little bit carefully. Let us look at both the cases. So let us look at the first case which we described just now. We have 2 points, x and x plus 2 points, and flow in is higher than flow out. If that is the case, we have J at x plus delta x minus J at x divided by delta x. This is our del J by del x, and this is going to be negative number. This is going to be less than 0. So this is, let us say, minus some number, minus 3. This is going to be some negative number. If you look at the other case, which is, let us look at this case where you have this two location, x and x plus delta x, and this is J at x and J at x plus delta x, and the flow in is less, flow out is more. If this happens, the concentration of protein here will deplete. The protein's molecules here will go away, and this concentration here will decrease, because all of these are going out. A lot of molecules are going away and only few are coming in. So if this is greater than this, J at x plus delta x is greater than Jx, so this will be a positive number by delta x, which is del J by del x. This will be greater than 0 because the flow here is more than the flow here. Therefore, this will be a greater than 0. Let us say this is going to be 5 or some bigger number. Now, if we write our diffusion equation, which is saying that dC by the change in concentration here is equal to this difference. So the claim we are making is that del C by del T is minus del J by del X. This is the claim that we are making. If this is the case, in the first case, if del J by del X is less than 0, then that would imply that del C by del T will be some positive number, 3 or some. If this is less than 0, this will be negative. Negative times negative is 3. Positive, it will be some positive number like 3. This would mean that C increases with time. C will increase with time at this location. If del C by del T is negative, if del J by del X is positive, del C by del T will be, this is positive, this is negative, this will be less than 0, that is del C by del T will be some negative number, negative 5. This would mean that C will decrease with time. C at that middle location will decrease with time. So, if del J by del X is greater than 0, C will decrease with time. If del C by del T, which is del J by del X, is less than 0, C will increase with time. So, this is something that you can check here. Here, in this case, del J by del X is negative. So, that means here C will increase with time, because the concentration here will increase, more are coming in. So, the concentration here will increase. Here, the concentration will decrease because del J by del X is positive and less coming in, more going out. So, I want you to check this and test yourself and convince yourself that this equation will indeed give us what we are wanting. So, what we want is the phenomenon of diffusion, which is basically description as a flow J, and this by demanding, if you take a point and if we discuss the flow which is coming in and the flow that is going out, if we balance those flow, del J by del X, how much is that flow itself changing? So, that is at any point in the cell, what we are worried is take any point in the cell, at any point you look at the flow that is coming in and the flow that is going out. So, J here and J here, this del J by del X, depending on this value of del J by del X, if the del J by del X is 0, this means whatever comes in goes out. If del J by del X is positive, that means this flow is more than this flow. If del J by del X is negative, this flow is more than this flow. So, the claim is that minus del J by del X is del C by del T. So, this is the understanding, and this understanding is just simply coming from take any location X, and around that you will look at the flow which is coming in and going out, and by studying this, considering 2 points very close to delta X away, we can discuss and write down such an equation. So, this is possible. Now, if we already know also that J is d minus d del C by del X. If you just take the magnitude, this is the magnitude and x cap is the direction. So, the magnitude is minus d del C by del X. So, if I substitute this J here, so let us substitute this J in this equation. So, I will get, so I am going to substitute this J here. So, do substitute this, and if I substitute, what do I get? I will get that del C by del T is equal to minus del J by del X. J is equal to minus d del C by del X. I substitute this J here, I will get del C by del T is equal to minus d, and there is a minus, so it will become plus del square C by del X square. So, this is the equation we will get, and this is the another form of diffusion equation. Del C by del T is equal to d del square C by del X square. So, this is the form of the diffusion equation. So, this equation is called the diffusion equation. This is called the diffusion equation. which says del C by del T is equal to d del square C by del x square. The equation is same as this equation. Equation is exactly the same. I just substituted this j here and I got this. I wanted to convey just one more thing about we discussed the magnitude. I also wanted to talk about direction. So, in vector analysis, if you have two vectors, del is a vector. This del vector is x cap del by del x is in one dimension. Del vector is del by del x x cap. And another vector we have is j. Now, if you want to get and we know that dc by dt is a scalar. This is a scalar, and therefore, this is c is not a vector, t is not a vector, this whole thing is a scalar. Therefore, we want to get a scalar here also. So, making a scalar is called a dot product. So, if I find a product between this and there is something called a dot product in mathematics, which is basically del dot j. Del dot j is dot product. This means that this dot this. So, this will have a component, let us call it j magnitude x cap. So, j magnitude, let us say this j is j magnitude x cap. So, the product is this and x dot x will become 1. So, this is nothing but del j by del x. In one dimension, this will be just del j by del x. So, this is the del j by del x that we are writing here. In other words, the diffusion equation or the continuity equation is actually del c by del t is equal to, in the vector form, it will be del c by dt is minus del dot j. This is the dot product. I would want you to understand this by studying the dot product of vectors, which we have studied a little bit in school. So, this j is a vector, del is a vector, the dot product of this will give this. Now, here you substitute the vector j is equal to minus d del c, where del is a vector. If I substitute here, I will get what we just discussed, del c by del t is equal to plus d del square c. Del square is the second derivative. So, this is the diffusion equation that we are saying. In 3 dimension, the del square will be x, y, z will be there, and in 1 dimension just x will be there. So, let me write this in 3 dimension and summarize what we did so far. So, if I write this in 3 dimension, del c by del t will be equal to d del square c by del x square plus del square c by del y square plus del square c by del z square. This is the equation in 3 dimension. In 1 dimension is just del c by del t is equal to d del square c by del x square. In 2 dimension, so this is in 1 dimension and this is in 3 dimension. And 2 dimension, there will be only 2 terms. So, the bottom line is that if we have a flow, if we think about flow, and if we take any point and look at the flow that is coming in and the flow that is going out, how the protein molecule flow at any point x, account for whatever coming in and account for whatever going out, and the difference between them will give us the concentration change with time, and that will give us this diffusion equation, and that will give us this equation which is called the diffusion equation. So, this is the diffusion equation in 3D or 1D. So, with this I will stop this lecture and continue in the next class. Bye.